Hello student, once again welcome to honors classes. Today is timing for the final revision for your biology question paper. So today in this my video I am going to give you some most important means probable questions from zoology and botany section. If you go through these answers, then at least 90% we will get common from these questions. Question may not be directly like this, but answer should be this. If you go through all the conception of this answer, then definitely you can attempt all the answers. So first question, write the name of different species causing malaria in man. Describe the life cycle of malaria parasite in brief and name the infective stage of malaria. So these are the first I am giving you name of 4 malaria parasite. In question paper you get may 1, 2, anything, but I am giving you 4, and this is the life cycle of malaria parasite. You just go through this diagram and 5 marks question come. With this diagram you just write explain in words also 2 3 line, but if only 3 marks question come, you just draw the diagram properly, you will get full marks. So this is the diagram of life cycle of malaria. Next, describe the immune system of body. So what is the different definition of immune system? I am giving the answer for 4 marks question. But if 3 marks question come or 2 marks question come, you just shorten the answer. So this is the definition of immune system of body. Next, immune system is of 2 types. So both types I am mentioned here in question paper. You get also write short note on cell mediated immunity, like that also you can get the question. So you just go through the immune system of your human body. Next, what is ecological pyramid? Explain different types of pyramids with necessary diagram. So these are the ecological pyramids, and here I am giving you the diagram also. If 5 marks question come with this diagram, you just as definition you just write one means description also. Next, what is the genetic code? State the principal feature of genetic code. I have already explained these answers in my previous video. Here also I am just giving you the important topic through which you should go for your before your biology exam. So this is the answer for genetic code and features. What is autoimmunity and name any two autoimmunity disease? So here I am giving autoimmunity disease. Here in question paper it has came as 2, but I am giving you example of 4 autoimmunity disease. What is DNA fingerprinting? Explain the process. So this is the definition of DNA fingerprinting, and these are the steps. If only 2 marks question come, you just write the heading whichever I have write in green letter. But if 5 marks question come, you have write as it is like this. And in southern blotting technique is also important. Sometimes short notes come from southern blotting technique also. If you go through the DNA fingerprinting, then southern blotting technique also come under this. It is a part of means DNA fingerprinting. Next, this is the diagram, and if only 2 marks question come with the definition, you just draw the diagram. No need to explain the steps. Describe the process of translation of genetic code. So this is what is translation. First I am giving you the definitions, then these are the steps. These 4 are the different stages of translation process. Next, describe the methodology involved in the Human Genome Project. So HGP in short form, we will get question like that also. So HGP means Human Genome Project. And this is the what is the goal of Human Genome Project? These are the goal of Human Genome Project. What is the causative organism of pneumonia? How a healthy person acquired pneumonia, state its symptoms. So first is the definition causative organism, then how pneumonia may be caused, and these are the signs and symptoms of pneumonia. Describe the various means of preventive and control of drug and alcohol abuse. So these are the, here I am giving the answer for 5 marks question. If 2 marks question, you just write 3 points only. Explain the Southern blot hybridization technique in DNA fingerprinting. Just few minutes ago I have told you it is a part of DNA fingerprinting. So if separately Southern blot hybridization comes, so you just write the definition and you draw the diagram only. You will get full marks. Describe the different stages of gene regulation. Explain lac operant model. So first is the definition of gene regulation, and 3 main level at which gene regulation are listed below. This is a 3 main level. Sometimes only question come write that 3 different level, then you have to write this level. Next, this is the description of lac operant model. So this is the mechanism of lac operant model. I am giving you everything about lac operant model. So if 2 mark question come, you just write the first para. If 5 marks question come, you have to write the mechanism also. Write the possible causes of cancer, write about its detection and diagnosis. So possible causes also given and detection also given here. Explain the codominance with example. So it is the Definition of codominance. Example. What is sexually transmitted disease? What are the main symptoms of AIDS? Discuss the method of prevention of AIDS. So first, definition of sexually transmitted disease. Early symptoms of AIDS are given here. Then prevention of AIDS are given here. Next, DNA is a genetic material. Explain the statement with the help of Griffith experiment. So this is the Griffith experiment. Already I have explained in my previous video. Still here I am just giving you for your revision only. Next, what is the multivoltine? And write the scientific name of two silkworm of Assam with their host plant. So this is the definition of multivoltine, and these are the two silkworm found in Assam. In local language, they are known as Muga silkworm and Airy silkworm. Next, write different types of RNA polymerase found in nucleus. So these are this is the RNA polymerase. And first is the what is no if question come, write notes on RNA polymerase. Then you have to write the point up to point number 4. Then types of RNA polymerase. These are the types of RNA polymerase. RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Sorry. RNA 4 and RNA polymerase 5. So these are the different types of RNA polymerase. Write the functional aspect of HGP. HGP means also Human Genome Project. In a few minutes ago, we have discussed in goal of Human Genome Project. These are the functional aspect of Human Genome Project. What is endemic animal of Assam is presently listed in Red Data book. So you have to write hologibbon. Hologibbon. Describe the process of flow of energy in different trophic level of an ecosystem. So these are the answer for this. You have to draw this diagram. You will no need to explain in parawise. You just draw the diagram. You will get full marks. And if 5 marks question come, then you just write the description also. Explain the greenhouse effect. So this is the explanation of greenhouse effects. Then write the 3 law of inheritance, law of dominance, law of segregation, and law of independent assortment. Single law may also come, but if you go through the all the 3 law, one question you will definitely get. Describe the pedigree analysis. This is the definition of pedigree analysis. Pedigree analysis question has last time come as long question. So this time only definition may come. Various chromosomal disorder, genetic disease. So these are the what is chromosomal disorder disease. First definition is given. And here I am giving you explanation of 3-4 chromosomal disorder, Down syndrome, like that. So shortness may come from any one of the disease also. Here I am giving Turner syndrome, such like that. I am giving you the explanation of 4 symptoms. Discuss various assisted reproductive technology used to solve the infertility problem. So this is the first definition of ART is given. Then the various ART, various types of ART is explained here. IVF, GIFT, ZIFT, like that. ICSI, these are the different methods. Write the structure of ecosystem. So this is the structure of ecosystem. So if you draw the chart, then also you will get marks. Describe the mechanism of transcription. So translation I have discussed, and this is the transcription process, and this is the diagram for transcription process. Then PCR technique. So this is the definition of polymer chain reaction technique. Next, important questions for botany. What is parthenogrammic fruit? How can the formation of such fruit be induced? So this is the definition of partner and how it should be induced. What is a SCP? Means single cell protein. Definition given here. And sometimes question may write to algae which are used in SCP. Then here, that's why I am giving you the name of the algae here, and name of the fungi also I am giving here. So if question may sometimes come, may also just write name of 2 algae which are used in SCP. Then out of these 3 you just write 2, or if fungi name is asked, then you write from here. What is plasmid? Write the main aim of biotechnology. So plasmid definition is also given, and aim of biotechnology. What is genetic engineering? What are the major steps of genetic engineering? First definition, genetic engineering or recombinant DNA technology is same thing, you know. So first para definition is given, then what is gene cloning is given here, and then the steps of gene cloning is given here. Why meiosis is essential at the time of sporogenesis? Describe the development of male gametophyte in angiosperm. So first, why sporogenesis? Meiosis is important given here. Then this is the sporogenesis. This is sporogenesis from this is from pollen mother cell to how the pollen grain is formed. This is first is divided two, then four cell, and in there is a generative cell and there is a pollen tube nucleus. And in the next step, this is from the pollen grain. Finally, one vegetative cell and generative cell is formed, and finally pollen from through the germ pore tube nucleus, germ tube is formed, and then two male gametes are formed. So these are the diagrams. Discuss the process of development of three-cell pollen grain from microspore mother cell with the help of a diagram. So from microspore mother cell, how pollen grain is formed, this is the given. This diagram, so I am given here. And this is the explanation. If 5 marks question come, then also you have to explain. Otherwise, you just write the 2-3 lines, one para and 2-3 lines of para and diagram, you will get the full marks. Survival of the fittest, explain with the help of Darwin's theory. So these are the answer for this question. Write briefly the chemical origin of life on Earth. So this is the answer for this question. These answers already I have given you in my previous video. Here still I am giving here once upon for your revision. What is double fertilization? Describe the process with diagram. So fusion of one male gamete with an egg cell and another male gamete with secondary nucleus is called double fertilization. And this is the explanation of double fertilization, and this is the diagram of double fertilization. Explain the process of embryo development in dicot plant. This is very important for this time. So this is the embryo development process, and this is the diagram of the embryo development. What is the somatic hybridization? Give an account of necessary steps on the method with example. So this is the definition of somatic hybridization, and where it is in tomato. Example, best example is tomato. What is the population growth? Write an account on the exponential growth model of population. The difference between the birth and the death rate is denoted by population growth. And exponential growth model, that answer is given here. Next, post fertilization state often changes in an embryo sac. So this is the explanation for that. What is GMO? How they are useful for us? GMO means genetically modified organisms. And these are the answer. Discuss the method of breeding for disease resistant crop. Method for plant breeding disease resistance are, these are the 4 methods are given here, and this is the explanations are given here in one line. Write on molecular diagnosis. So this is the definition of molecular diagnosis: to identify a particular sequence of nucleotide in a mixture of nucleotide. Explain megasporogenesis. So this is megasporogenesis. Previous one megagametogenesis, and this is megasporogenesis. And rule of microbes in biogas production. So here the rules of microbes mean here rules of bacteria, rules, everything is given here. What is PCR? How amplification of DNA is done by using PCR technique. This is also important, very important. So these are the different steps. This is given for 5 marks. If 3 marks come, you just write one step. What is GEAC? How it controls the genetic engineering research? GEAC means Genetic Engineering Approval Committee, and how it control the genetic research is the two-way. Discuss the modern synthetic theory of evolution. This is the modern genetic theory of evolution. Write about the principle and function of ELISA technique. So this is the principle and function of ELISA technique. So dear student, you please go through these answers, and if you go through properly, go through this answer, any type of question may come, 1 mark, 2 mark or 5 marks, you can write from this answer. At least 95% of question answers will be come from these answers. So please go through properly with this answer, and all the best for your coming exam. And till then, thank you.